So we've been talking about sodium, potassium moving into and out of the cell, but why do they move in and out of the cell? Why does our body want them to move in and out of the cells? Does our body want them to move in and out of the cells? Because they will facilitate a change in voltage within the cell. So if we look at this cell right here, this cell is polarized. That is to say, and this is how cells are when they are at rest. Negative on the inside, positive on the outside. That's actually showing some sort of polarity, and it's not a polar molecule, but it's just polarity once again is exhibiting two different characteristics on different ends of whatever it is, but in this case it's the cell, negative on the interior of the cell and positive on the exterior of the cell. And once again we refer to this as a polarized cell, and this is how resting cells, the state of resting cells, they are polar. So to be very clear, most cells have a polarity, but not all cells can change their polarity. When a cell can change their polarity, like muscle cells, nerve cells, and a host of other cells in the body, I tend to refer to that as becoming excited. If a cell can change its polarity or its voltage, it's referred to as excited or depolarized. How does a cell depolarize? Well, that's by sodium moving into a cell. And it can happen by another ion known as calcium moving into a cell. And it can happen a number of ways, but probably the most common ways with the influx of sodium. So we've talked about the influx of sodium, sodium moving down its electrochemical gradients. But why do we want that to happen? Well, we want it to happen so the cell depolarizes. If enough sodium comes in, the interior of the cell turns positive, and since the outside is positive, it's referred to as depolarized because there's no longer a polarity between the inside and outside of the cell. But I still haven't said why we want a cell to depolarize. A depolarized cell causes things to happen in a cell. A muscle cell, when it depolarizes, is going to contract. So the depolarization of a muscle cell facilitates contraction. The depolarization of a neuron facilitates the release of neurotransmitters. I realize we haven't gotten to neurons yet, but we will. And what we'll talk about in a moment here are pancreatic beta cells which release insulin. If a pancreatic beta cell depolarizes, it releases insulin. So depolarization is due to sodium influx, the movement of sodium into the cells, causing the interior of the cell to turn positive. Now, we don't want cells to remain depolarized because if a cell is depolarized, it cannot depolarize again. So for a cell to depolarize again, it must first go back to its resting state, the polarized state. And that's what's known as repolarization. This cell is repolarized. It should look and does look exactly like this first cell, but the process that brings us back to this state, the polarized state, is known as repolarization. And that is caused by potassium efflux, potassium leaving the cell. To be clear, I realize I don't have brackets connotating concentration around sodium and potassium, but from here forward, if I don't have brackets around sodium and potassium, please understand that they are there. They're just invisible, that we're always talking about concentration. So the efflux of potassium causes repolarization. The influx of sodium causes depolarization. And the depolarization of a cell will elicit some downstream activity. Okay, so a great example of the utility and need for depolarization is the release of insulin from pancreatic beta cells. So to be clear, right here in blue, we have what's known as a secretory vesicle. And this is containing, or I should say secretory vesicles contain some sort of molecules that will eventually be released from the cell. So this is a secretory vesicle. these blue beads within the secretory vesicle are insulin. So insulin is a hormone that regulates glucose levels in our body. Insulin is released from the pancreas. The pancreas is an organ that sits behind the stomach, right next to the duodenum, which is the first part of the small intestine. And within physiology, in my opinion, by and large, it doesn't matter that you understand where specific organs are, but to be clear, the pancreas, which looks like this, sits behind or posterior to the stomach. And the pancreas is both an exocrine gland, that is to say, it's secreting fluids and chemicals and digestive enzymes into the digestive tract, but it is also an endocrine organ. The endocrine system is a system that releases a signaling molecule, or signaling molecules, I should say, into the bloodstream. Insulin is released from beta cells of the pancreas into the blood. Also, and the beta cells are sequestered within regions within the pancreas known as the islets of Langerhans. So as we see here in the pancreas, this region right here within the pancreas that I have outlined in green, I'm not sure you can see the green, is the islets of Langerhans. And the islets of Langerhans contain a number of cells, but two very important cells, beta cells and alpha cells, that help regulate our glucose levels in our body. Beta cells release insulin, alpha cells release glucagon. Insulin is released when we have high levels of glucose in our blood that we need to get throughout our body and into our body cells, so our cells can utilize glucose to make ATP. So when we have high glucose levels, which is generally after a meal, dare I say always after a meal, we have high glucose levels. The beta cells, when the beta cells detect high glucose levels, insulin is going to be released, allowing our downstream cells to utilize that glucose. And we'll talk about how our downstream cells utilize insulin to allow for glucose to move into those cells, which is known as glucose uptake. But first, we need to talk about how insulin is actually released from beta cells, the mechanism that allows insulin to be released. And I think this is a wonderful example of everything we've talked about: the phospholipid bilayer, the voltage across cell membranes, the movement of sodium and potassium and other molecules, different types of channels. So this is a pancreatic beta cell right here. This whole cell is a pancreatic beta cell. Once again, this is a secretory vesicle that contains insulin. And we have a number of channels in this cell. Right here, we have a glucose leakage channel. There's a number of different types of glucose channels in the body that we will talk about over time. I'm not going to tell you what type this is because I don't care at this moment. But this is a glucose leakage channel. That is to say, it has no gate and it is always open. It allows glucose to move into the cell. Right here is a sodium leakage channel. It's not a gated channel. It allows sodium to move in whenever it wants, when there's a higher concentration of sodium outside the cell than inside the cell. So to be clear, when we talk about gated channels, if you open up that gate, it's like opening up floodgates and things just come in like a flood. A leakage channel is like a faucet that's just kind of dripping, dripping, dripping. And in this case, with a sodium leakage channel, it's just dripping sodium into the cell. Up here, we have a very unique channel known as the potassium ATPase channel. It's also known as the potassium pump, because anytime we see the word ATPase, we can also use the word pump, and it's also suggesting it's an enzyme that's going to break down ATP. So once again, this is a potassium ATPase channel. If the potassium ATPase channel is open, we should already know what potassium is going to do. That is to say, potassium is going to leave the cell down its chemical gradient, because that's what potassium does. This right here is a voltage-gated calcium channel. Now, we haven't talked about calcium yet, but calcium is a cation, very similar to sodium, but it has 2 positive charges rather than 1. This is a voltage-gated calcium channel. There's a much higher concentration of calcium outside of cells than inside of cells. So if this gate opens, calcium is going to move in down its electrochemical gradient. It has a positive charge, 2 positive charges, just like sodium. Sodium has 1 positive charge, but they are both cations. So once again, glucose leakage channel, then we have the sodium leakage channel, the gated potassium channel known as the potassium ATPase, and the voltage gated calcium channel. So what we have after a meal, when there are high levels of glucose in the blood, glucose moves into the cell. When there's high levels of glucose in the blood, there's an influx of glucose into the pancreatic beta cell. And when glucose is in the cell, it's going to react with oxygen. When glucose reacts with oxygen, what happens? If you know, like and subscribe and send a message telling me what happens when glucose reacts with oxygen. And if you said it creates ATP, you are correct. Glucose plus oxygen creates ATP plus the heat, water, and carbon dioxide. As we move through physiology, you should always have that equation of cellular respiration at the forefront of your mind when things are happening, because that's super uber important to human physiology. So to be clear, when there are low levels of glucose, glucose isn't moving into the cell, or minimally so. So when glucose is not moving in, so let's just get rid of glucose, we have sodium leakage moving into the cell. But that's going to be offset by potassium. That is going to be offset by potassium leaving the cell. So the cell is not going to depolarize. Sodium comes in, but potassium's leaving. So the cell is going to remain polar, negative on the inside, positive on the outside. And this voltage-gated calcium channel is not opening up. But when we have high levels of glucose, this is what happens. Okay, so what happens when there is high levels of glucose immediately after a meal? We have a large influx. Of glucose into the cell. And when glucose moves into the cell, glucose is going to react with oxygen to make ATP. That ATP is going to bind to the potassium ATPase channel and close that channel. Now, to be very clear, I love this example because generally we talk about ligands opening up gates. Gated channels generally open when a ligand binds to it, but you'll find out there's a lot of variation in the body. And in this case, when ATP binds to this channel, it closes the gate. And when that gate closes, potassium, which is now super sad because it can't move through that channel anymore. Keep in mind, sodium is still moving through its leakage channel. So with sodium moving through the channel and potassium no longer able to leave, the cell starts to depolarize. And that depolarization event is going to open up the voltage-gated calcium channel. We're going to get an influx of calcium down its electrochemical gradient into the cell. And one thing you may have noticed is that moved this secretory vesicle to the perimeter of the cell, where we now have the process known as exocytosis, which releases this insulin into the bloodstream, allowing for our downstream cells to utilize glucose. So to be clear, high concentration of glucose moving into cells creates a bunch of ATP that binds to the potassium ATPase channel. When that channel closes, potassium can no longer leave the cell. Sodium is still coming into the cell, because sodium is coming in and potassium is no longer leaving, the cell starts to depolarize. And that depolarization event opens up this voltage-gated calcium channel. Calcium channel opens, calcium moves into the cell and facilitates what's known as calcium-induced exocytosis, which in this case releases insulin. So on the face of it, this example of the release of insulin from pancreatic beta cells may seem complex and difficult, but if you understood the previous lectures on the plasma membrane, the different gated channels, voltage across membranes, this is just incorporating all those concepts. One thing I do want to keep in mind, when I do my drawings up here, these are not real life drawings. That is to say, I've drawn one potassium ATPase channel where there could actually be 100. And I've only drawn one sodium leakage channel where there could be 1,000. So this is very simplified, and this is the release of insulin from pancreatic beta cells.